Hello everyone, today I would like to talk about the variability of foreshore and nearshore marine animals on a Cobble beach in Nova Scotia. For my introduction part, the coastal zone plays an essential role in the development of our country. It has shaped the Atlantic Canada's historical, ecological, cultural, and industrial growth. Nova Scotia, where the experiment was conducted, is economically and sociologically important Atlantic province, situated in Mi'kmaq, the Mi'kmaq people's ancestral region. Coastal habitats are able to accumulate and transport nutrients into neighboring ecosystems, thus contributing to the environmental stability. It's really important to consider that the nearshore area is vulnerable to different natural factors since it is a transitional zone directly affected by human activities. Therefore, species richness and diversity depends on the coastal heterogeneity. Thus, it is of immediate interest to study the variability of foreshore and nearshore marine animals to impact the influence of changing environment on species distribution patterns. Our research question aimed to find the difference in the diversity of marine animals between the foreshore and the nearshore of a Cobble beach at a public area and boat launch in Nova Scotia, Canada. The answer to this question would uncover the effect of the environment on marine animals' distribution patterns. To create a valid model for comparison, a null hypothesis was designed that allowed to make statistical calculations. The null hypothesis stated that there would be no significant difference between the diversity of marine animals on the foreshore and nearshore of the Cobble beach at public area and boat launch in Nova Scotia, Canada. Field Experiment 2 was held at the public area and boat launch at 6655 South Street, Halifax, Nova Scotia Province, Canada. The exact location is marked red on a Google Maps screenshot in Figure 2. The postal code of the site is NS B3H4L3. The experiment location site is located in close proximity to Dalhousie University. The photos of the field experiment 2 location site are shown in Figure 3 and 4. Before developing the research question, the studied area was analyzed for the abundance and diversity of different marine animal species. The examination proved the presence of numerous marine animals. Halifax City Hall was contacted regarding the permission to perform the experiment. No permissions were required as the beach surroundings were not disturbed during the experiment. The experiment was planned to start at 5:05 p.m. on May 19, 2022. Foreshore environment analysis was begun first. It would happen a low tide at 6:13 p.m., according to Fisheries and Oceans Canada, 2022. After the low tide, the nearshore experiment was begun and should take less than 1.5 hours since. The total duration of the experiment should span on experimentally 4 to 5 hours, measuring foreshore environment first, before low tide can keep safe. Materials needed for the experiment included a 3 meters measuring tape, plastic sticks to make a quadrant, a waterproof notebook, pencils, and an iPhone camera. The total cost of the materials was approximately $40, excluding the camera. The experiment was designed to compare the diversity of the marine animal species inhabiting the nearshore zone and the foreshore zone. So, firstly, to measure the diversity of marine animal species, a 30 meters measuring tape was placed parallel to the waterline on the middle foreshore at 17:05, as shown in Figure 5. This is a field photo. Place the first quadrant at 1745 and the random distance along transect was 2.79 meter. Place the second quadrant at 1750 and the random distance along transect was 4.99 meter. Place the third quadrant at 1752 and the random distance along transect was 10.39 meter. Place the fourth quadrant at 1755 and the random distance along transect was 14.99 meter. Place the fifth quadrant at 1759 and the random distance along transect was 17.79 meter. With the help of a random number generator, 5 locations or replicas were chosen along with the measuring tape. Then, waiting for the low tide spent on 40 minutes. The low tide happened around at 1813. After the low tide, a 3 meters measuring tape was placed parallel to the waterline on the middle near shore at 1814, also shown in Figure 5. Place the first quadrant on the nearshore zone at 1815, and the random distance along transect was 2.54 meter. Place the second quadrant at 1818, and the random distance along transect was 5.89 meter. Place the third quadrant at 1820, and the random distance along transect was 8.18 meter. Place the fourth quadrant at 1825, and the random distance along transect was 11.92 meter. Place the fifth quadrant and 1827, and the random distance along transect was 13.19 meter. With the help of a random number generator also, 5 locations or replicas were chosen along with the measuring tape. Totally, spent on last half hour on nearshore zone. For each quadrant, photos were taken by iPhone. Data was recorded and collected in a notebook, including measuring time, random distance along transect in meters, marine animal species names, and the number of these species. Finally, the Cobble beach was cleaned up. The experiment materials were taken away. To count the number of marine animal species of foreshore and nearshore quadrant photos, physics programs was used. To evaluate the significance of the results, competent Shannon equilibrity index was calculated, which measures the evenness of species in a community. The equation is shown on the slide. Shannon equality index is represented by H. Pi is the proportion of the alpha species in the natural logarithm of Pi, and S is the number of species in a community. The higher H value means the higher diversity of marine animal species in a community. The lower H value means the lower the diversity. When H equals 0, means the zone only has one species. T-test was used to compare the 5 values of H under foreshore condition with the 5 values of H under nearshore condition. Each quadrant should have one value of H. The alpha value of this analysis is equal 0.05 if the p-value from the t-test was larger than 0.05. Then we failed to reject the null hypothesis as there was no significant difference between the diversity of nearshore and foreshore marine animals. If the p-value from the t-test was smaller or equal to 0.05, then the null hypothesis can be rejected because there was a significant difference between the diversity of nearshore and foreshore marine animals. There are 3 assumptions we need to check for the t-test. The first one is called independence assumption. This can make sure the populations you are testing are independent of each other. The second one is called equivariance. This can make the variance of each population to be approximately equal. The third one is called normal distribution. This can check your calculated data whether normal or not. This is my results. The results of the field experiment 2 are illustrated in the figure 6 and 7. The graphs indicated the clear difference between species diversity on the nearshore and foreshore. Periwinkles predominated in the nearshore species groups. Various barnacles constituted the majority of marine animal species found on the foreshore. The average number of dogwills, blue mussels, and Baltic magmas per 400 centimeter square did not show drastic fluctuation. The mean age for nearshore marine animal species number was 0.56, and the mean age for foreshore marine animal species number constituted 0.53. Based on the mean age for nearshore and foreshore, mean age value of nearshore was 0.03 greater than mean age value of foreshore. Thus, we can know marine animal diversity of nearshore was similar with the foreshore. Through t-test to compare and analyze age values of these two zones, we got its t-value was 0.218, degree freedom was 4, and the p-value was 0.838. Since its p-value was larger than 0.05, we failed to reject the null hypothesis and concluded there was no significant difference between the diversity of marine animals in the foreshore and the nearshore of the Cobble beach at the public area and boat launch in Nova Scotia, Canada. This followed the three assumptions: independence, equivariance, and normal variance for the t-test, which was normally distributed. This is determined through Shapiro test. There was not a transformation done to normalize the data. The results indicated that although diversity is not significantly different, there are very different distribution patterns between nearshore and foreshore regions of this Cobble beach in Halifax, Nova Scotia, indicating that different environment influences species richness and abundance. These findings correlate well with the data obtained from the literature analysis. It was shown that the climate region and distinct habitats influence the nearshore fish assemblage structures. A study by Tuya et al. In 2019 demonstrates that taxonomic diversity of fish varies depending on the habitat. Difference in sea surface water temperature and salinity impact crustacean assemblages, according to Kondo et al. With an 83% prediction success rate, fish assemblage patterns in mangroves might be connected by physical-chemical factors. The submarine groundwater is reducing turf species richness. Furthermore, diverse fish assemblage were found in foreshore bar and nearshore true environments. Similarly to the competent results, mollusca were the most varied and prolific group of creatures in Samianiban. I think the changes in species abundance could be attributed to secondary influence factors. For example, there is the possibility for mutual subsides between marine and terrestrial ecosystems in marine nearshore habitats. It was shown that barnacle recruitment increases linearly with the increasing upwell frequency. Additionally, microplastic is found in nearshore species and can decrease their survival rate and abundance. Finally, the presence of herring spawn was probed to cause an increase in macrophages and invertebrate populations, which might also impact the species abundance in the marine environment. The present study includes several limitations, such as small sampling number, short sample collection time period, and data unavailability on physical-chemical parameters of foreshore and nearshore environment. Future experiments could include 10 to 15 samples in sites, several weeks or months of monitoring, and water salinity, pH, and temperature measurements. It would be also crucial to include of indigenous advisory community, use traditional knowledge gathering techniques, and interpret data with the knowledge holders according to the two-eyed seeing principles. In conclusion, study explored the distribution patterns of marine animals on the nearshore and foreshore of the Cobble Beach in Nova Scotia. The results proved the variability of marine animals' abundance depending on the habitat. More experiments are needed to determine the cause of the differences in species frequency of occurrence. Thank you for your listening. Thank you for my professor Isabel and the demonstrator Alan. This part is for my references. Thank you.